All right. So today, this morning, we have a session on phytoplankton physiological engines of biogeochemical models. And I just wanted to provide a very brief introduction on behalf of my co-sponsors, Phil Boyd, Scott Toney, and Charlie Stock. This is a metabolic map of Prochlorococcus marinus, which is a globally ubiquitous phytoplankton with perhaps the simplest genome. And you can see it's already wildly complex. So we want to have robust and data-constrained models that describe photosynthesis, multiple nutrient limitation, co-limitation, stoichiometry, and mixotrophy. And you can see that that's going to be quite complicated. So the challenge really is, how is it possible to represent the population dynamics of a complex organism in a variable environment? And so that's really what this session is about. There's been tremendous progress in this area in recent decades. So I don't want to say that there isn't progress in this area. In fact, there's quite a lot. All of these models reflect this fundamental tension. We want to have a physiologically reasonable model that has good data to undergird it. But we don't have perfect knowledge of a lot of these processes, and we want the models to be computationally tractable and feasible. So today, what we really want to do is talk about some of the recent advances in this area. So these kinds of models are built into global biogeochemical models that are very important for looking at ecosystem dynamics, the carbon cycle, and climate impacts of marine ecosystems. And we really want to see what recent progress there's been in this area. In particular, we're going to talk about photosynthesis, variable stoichiometry, multiple nutrient limitation, and mixotrophy. And we really ask, what are we doing well? What can we do better? And where might models go in the near future? We have 4 really spectacular talks, in my opinion. We'll start off with Richard Geider talking about modeling acclimation and adaptation of photosynthesis in microalgae and cyanobacteria. We'll have Aaron Bertrand talking about phytoplankton physiological responses to simultaneous nutrient or micronutrient limitations and multiple stressors. Adam Martini will talk about understanding and predicting the regulation of ocean, sea, N, P, and export production. And then finally, Stephanie Dukevich will talk about physiological diversity matters, a modeling perspective, and she'll talk about a range of different modeling strategies that people have taken recently. So I hope this provides you just a very brief introduction about the kinds of themes we want to talk about. And with that, I would like to transition to the people who really are pushing this all forward, starting with Richard Geider.
Is this going to work? Can you hear me? Yes?
You guys hear?
Okay, thanks.
Here's your clicker.
Okay. Well, at the beginning of this session, you saw that very complicated model or representation of an organism. And yesterday, we heard a great deal about the very rich tapestry of interactions between evolving organisms and their environment. And what I'm going to do is actually focus on a very, very narrow range, just a few stitches in that tapestry, and I'm going to talk about the photosynthetic physiology of phytoplankton. The talk is organized into 2 parts. First, there's going to be a little bit of a history lesson, despite all of the mentioning of the word recent a couple of minutes ago. I'm going to give an overview of a model that links photosynthesis, respiration, nitrogen assimilation, and chlorophyll synthesis in phytoplankton. That was a model that was developed by in collaboration with Todd Canna and Hugh McIntyre, and then revisited about 10 years ago with a few tweaks with Oliver Ross. Then what I'm going to do is actually turn to some experimental work. I'm going to show you some work I've done with colleagues at the University of Essex on the acclimation of photosynthesis rates and photosynthetic proteins to nitrogen, phosphorus, and light limitation in one organism, in Amelia huxleyi. But I think that the responses we see in E. hux are not atypical, and I could draw on other data to show that. So what we wanted to do 20 years ago was to develop a model that was going to account for changing growth rate, changing carbon to nitrogen stoichiometry, and changing chlorophyll to carbon ratios, and account for that variability in response to light, into nutrient availability, and into temperature. And at the core of the model is the way in which we deal with the parameterization of chlorophyll synthesis. And the approach that we took is based on what we know about how plants and algae regulate the rate of chlorophyll synthesis. And they use a signal that's generated within the thylakoid membrane. And we can parameterize this signal in terms of what we know about a photosynthesis light curve. It's related to a concept that sometimes referred to as excitation pressure. There's an alternative hypothesis, which is based on the idea that phytoplankton will maximize their growth rate by changing the allocation of biomass between the pigments that absorb light and the catalysts that use this light energy for growth and biosynthesis. So there's another way of getting at this acclimation. For heuristic purposes, we can consider the phytoplankton cell to have 3 carbon pools. So it's a very, very simple cell. One of these pools is the biosynthetic apparatus, which we've assumed is a constant proportion of biomass. Then we have the light harvesting pigments and associated proteins, and an energy storage reserve. And those are variable. So a very simple structure. Under low light, the size of the light harvesting apparatus expands to collect more photons. And under high light, it contracts to avoid photooxidative stress. So those are the ideas behind it. Show you just one slide with equations. The model has a carbon budget, the difference between photosynthesis and respiration, a nitrogen budget between nitrogen uptake and remineralization, and then a chlorophyll budget, chlorophyll synthesis and degradation. But these equations are linked. So the photosynthesis rate depends on the chlorophyll and nitrogen status of the cells. The respiration rate depends on the rate of nitrogen assimilation, and chlorophyll synthesis also depends on nitrogen assimilation. And then this regulatory term, which takes into account the way in which the cells sense their environment. So for photosynthesis, we just have a photosynthesis light curve with a maximum rate depends on the nitrogen-carbon ratio is downregulated as nitrogen drops. And the initial slope depends on the chlorophyll to carbon ratio and increases in chlorophyll content. Increases, very straightforward. Respiration has a maintenance component and a cost related to nitrogen assimilation. Nitrogen assimilation is a saturated function of nitrate in the environment, again, with a variable maximum rate. Now the downregulation occurs as nitrogen increases rather than decreases. And then chlorophyll synthesis is related to nitrate assimilation, but the slope of that relationship changes depending on the light environment. We ran the model for steady state, and we can compare it with experimental results for 2 organisms. The lines represent the model output, and the symbols are the observations, and you can see we can capture the relationship between growth rate, nitrogen to carbon ratio, and also the effect of light. And we can also capture the relationship between the chlorophyll to nitrogen ratio and growth rate. So these are the steady state responses. The model also gives us the dynamics of the response, the kinetics of the response, which is really why I got interested in this in the first place. We only have to look at this lower panel here. If we go from low light to high light, the chlorophyll to carbon drops. From high light to low light goes in the opposite direction. You can see that we can capture the kinetics of the response as well as the steady state response. There's actually very little data available for us to to test and to confirm these things. As I said, there was an alternative explanation which is based on optimal allocation. And in this review paper, which was written a few years ago, my name was mentioned, so I thought I'd draw attention to it. And it was not mentioned in a positive sense. It was mentioned in the sense that these optimality models do a much better job. So I want to show you the figure from this paper. It plots the chlorophyll to carbon ratio, gets nitrogen to carbon for some nitrogen limited studies and for a light response. The data is from Laws and Bannister's 1980 paper, so it's real data represented by the crosses. Then the model that I've been involved with has the problem, according to this review, that we get a constant nitrogen-carbon ratio, a light limitation, whereas the data seems to show a different response. But we capture the response to nitrogen limitation. Oh, sorry about that. That wasn't supposed to happen. Yeah. So Rob Armstrong developed a model which has a break in it to account for this reallocation. And then Paolo and Ashleys actually do a very good job of capturing the light-limited part of this response. So this is the data for light limitation from the Laws of Bannister paper, and you can see that as you go from high light to low, nitrogen to carbon stays stable, that it increases, chlorofluorocarbon increases throughout. So consistent data with model. But I can show you another organism. This is some work that I did on Phaeodactylum tricornutum. No change in nitrogen-carbon ratio, chlorofluorocarbon increasing. So you get different responses for different organisms, and we might expect that we have to modify the model to do that. This is also some of my own data, so I can see I've also seen a decrease in the nitrogen to carbon ratio with increasing light. But in this case, in the lower panel, we can attribute this to the accumulation of energy storage compounds. So the contribution of carbohydrates to carbon is these open symbols. This is the residual carbohydrates after we extract the energy reserve polymers from the cells. So the decline in nitrogen to carbon is associated with the accumulation of these energy reserves. We revisited the model with Oliver Ross about 10 years ago, and that was because we had some experimental results in which we did a light shift. We had grown skeleton hemocaustatum at low light for a period of time, shifted it to high light, and then shifted it back to low light. This is the data for the nitrogen to carbon ratio, which drops in high light, the chlorophyll to carbon, which drops in high light as well. This is a fit with the original 1998 model with parameter values similar to those in there. We don't do a very good job of capturing this decline here. We don't capture this variability here. There's day-night variability in here, so the vertical strips represent either the light period or the dark period. And we wanted to capture that. The first thing that Oliver did was to actually tweak the model by changing one of the parameters, and we can now actually get this day-night variability in the chlorophyll to carbon quite easily. We get day-night variability in the nitrogen to carbon, but it still doesn't do a very good job with the data. So we now made this energy reserve pool explicit, separate from the rest of the biomass. This is just there as an indication that photosynthate goes through a low molecular weight pool to biosynthesis, or in high light to energy reserves. Then in low light, we can use those energy reserves to continue biosynthesis. A few little tweaks to the model other than that, and now we can capture the diel variability in the nitrogen to carbon, the response to high light and low light. We also capture the response to in the chlorophyll to carbon ratio. So a few little tweaks so we can get there. Now what I want to do is to turn to the second part of the talk, in which we did some experimental work into light acclimation in Amelia huxleyi. We measured photosynthesis rates. We also measured elemental composition, and we did some proteomics. In terms of photosynthesis, what we find is that in response to high light and low light, similar maximum rates, different initial slopes. So the kind of response that we have in the model. We then asked, how is the proteome of this organism responding to these changes in environmental conditions? And we used a non-targeted approach, came up with over 1,000 proteins that could be identified. And I'm only going to look here at some of the photosynthetic proteins. And what I've done in this figure is to break it up into several components. So the photosynthetic electron transfer chain, the chloroplast, ATP synthase, Calvin cycle enzymes, some enzymes involved in respiration. I'm plotting the contribution to the proteome in highlight versus the contribution in low light. And the dashed line is the one-to-one relationship, so it's no change. It's exactly the same in both conditions. And actually, you can see that there's not very much of a difference. Each of these points represents a different protein. For the photosynthetic electron transfer chain, there's less abundance of these proteins in highlight than low light. For the ATP synthase, these are different proteins within a macromolecular complex, so we expect them to co-vary. If they didn't co-vary, it would tell us that part of that complex wasn't being synthesized. So it's not surprising that the co-variability there, just a little bit lower in highlight than low light. Calvin cycle enzymes, again, not much of a change. But if anything, lower in highlight than low light. The thing that increases are the proteins associated with respiration. So if we lump now at the macromolecular complex level or at the biochemical pathway level, we can compare the low light to highlight proportions of the proteome. This is just the ratio. So for the electron transfer chain, it's about 50% higher in low light than highlight. For the ATP synthase and the Calvin cycle, it's maybe 15% or so. So as a response to light, not a big change in this component of the proteome. There is a component that did change, and that's with the light harvesting pigment protein complexes. There are about 70 or 80 different proteins that bind pigments that are involved with absorbing light in this organism. We detected all of the ones that are marked with either red or green. Red is a higher abundance in highlight, green is a higher abundance in low light. Again, if we now group all the proteins that we detected, the abundances, at these levels of these different groups, we have light harvesting chlorophyll fucoxanthin proteins 1, 2, and red. These are all involved in photosynthesis, and they're higher in low light. Then we have these other two groups which are involved in photoprotection, and they're lower in low light, higher in highlight. So these increase photosynthesis in low light. These provide photoprotection in highlight. So response a little bit more complicated, but it's not unexpected. We grew the same organism under phosphorus and nitrogen limitation. So in the center is the nutrient replete, in this case the growth rate. We use chemostats, so we get a nitrogen-limited growth rate, a phosphorus-limited growth rate. We see the kind of physiological responses that, again, are not atypical. Phosphorus-limited cells tend to be bigger, they have a bigger volume, bigger carbon content. They also contain more chlorophyll than nutrient replete, but that's associated with having more carbon. The C to N ratio is lowest under nutrient replete conditions. Lots of carbon accumulation under nitrogen limitation, a little bit less carbon accumulation under phosphorus limitation. Again, not an atypical response. And the nitrogen to phosphorus ratio is low in N-limited cells and high in P-limited cells. So everything as expected there. We also looked at how phosphorus and nitrogen limitation affect photosynthesis. And here what I'm doing is plotting the light-saturated photosynthesis rate against the growth rate. We're doing it for both net photosynthesis and gross photosynthesis. And so net photosynthesis, we get a linear relationship, and gross photosynthesis, we get a linear relationship with a little bit of an offset, so it's not a surprise. And really this correlation between maximum photosynthesis and the achieved growth rate is observed under other nutrient limitations as well. It has to happen if there's a feedback between growth and photosynthesis and a regulation of photosynthetic activity. In the model, we parameterize the maximum photosynthesis rate in terms of the nitrogen to carbon ratio. And although we could do this for the nitrogen-limited cells here, we try to do it for both phosphorus and nitrogen-limited. What we see is a divergence, nitrogen limitation up here, phosphorus limitation down here relative to the nutrient replete. So there's a little bit of a divergence there. As with the experiments under light limitation, we wanted to see what happened in the proteome. Before looking at the proteome as a whole, what I'm going to do here is plot the maximum photosynthesis rate, now normalized to protein. So this is total photosynthesis divided by total cellular protein. And you can see that it's high under the nitrogen-limited condition and low under the phosphorus-limited condition. And what this tells us, which makes a lot of sense, is that the nitrogen-limited cells use their proteins much more effectively for photosynthesis and growth than do the phosphorus-limited cells. We might almost think of the phosphorus-limited cells as accumulated protein as a nitrogen reserve, but it's an important nitrogen reserve because the proteins are all sitting there waiting to be used as soon as the cells can accumulate phosphorus. So they're primed to respond to phosphorus availability. It also tells us that we should think about regulation of the activities of these proteins as well as their abundances. We looked at the proteins that, again, were associated with photosynthesis. So now I've lumped all the proteins involved in photosynthetic electron transfer, looking at the nutrient-limited contribution to the proteome versus the nutrient replete. For both phosphorus and nitrogen limitation, again, the dashed line is the one-to-one, the no change line. And for photosynthetic electron transfer, I think you feel comfortable there's no change. So the proportion of the proteome associated with this is the same in all three conditions. They're all grown under the same light. And moreover, they don't diverge between phosphorus and nitrogen limitation. So very conservative. We look at the Calvin cycle enzymes, there's a lot more scatter. But again, there's no divergence between nitrogen limitation and phosphorus limitation in terms of their contribution to the proteome. And it's not wildly different from the nutrient replete. So, brings to the conclusions. We can parameterize downregulation of pigment synthesis using an index of excitation pressure. So going back 20 years there. The catalytic rate of the enzymes are regulated by nutrient and light status of the cells, so we need to consider regulation in here. Regulation is likely to be important in controlling how phytoplankton use light and nutrients, as is the allocation to these catalysts. Energy, organic carbon, and nutrient storage pools are important. And although most of the work that I've shown you was under steady state conditions, having these pools will allow the organisms to exploit episodic availability of resources. And I'm going to have to end the talk there, so I've run out of time. So thank you, everyone.
Time for questions?
Yes.
All right. In your plot where you looked at the photosynthesis normalized to protein biomass, you sort of said that on the phosphorus limitation, there was like a big protein pool that you're thinking of as storage for nitrogen. On the P limitation, that's why there was such a low ratio of photosynthesis activity to protein. But couldn't it also be that there's a lot of proteins when you're P limited that is allocated towards P stress, like alkaline phosphatase, P uptake, etc.? I assume you did. I think if I understood it right, you were doing proteomics as well in that experiment. So you should be. Did you see that?
Yes, actually, what we saw in all the proteomics experiments is that there were changes in the part of the proteome that responded to the stress. So under P limitation, there was lots of alkaline phosphatase. Still only a very few percent of the total spectral counts that we detected. There was also upregulation of some of the proteins involved in phosphorus transport. Under nitrogen limitation, there was upregulation of the proteins associated with nitrogen uptake. As I showed you for light limitation, there's a big change in those proteins involved in light harvesting. So when you think about it, is those proteins that are most important for acquiring a resource do change. In terms of nutrient uptake, most of those proteins are associated with the cell membrane. And the cell membrane is actually a small proportion of biomass. So in terms of the proteome of the cell as a whole, you get big changes in the membrane, but it's still a small compared to the total biomass. So yes, you get very pronounced changes in those proteins that are important.
So, Richard, one of the things Andrew didn't mention at the start was one of the origins for the idea of this session was the fact that many of the global biogeochemical models we use, many of the regional models we use, are still very much rooted in those ideas expressed in your papers from 97 and 98. And I think it reflects really the powerful blend of mechanistic simplicity that they created. The second half of your talk, you were sort of highlighting some of the new dimensions that may have been omitted in those original models, and some of the later speakers will also be kind of talking about these new frontiers. I was just curious what your thoughts were on some of the key pieces that you see now that had you to do it again. What would you add to those original models?
Well, I haven't been completely idle in the last 20 years. I have been distracted by other things, but I have given some thought to it. And I think that as soon as you get into multiple stressors and you want to include the potential for co-limitation between different nutrients, the models are immediately going to become much more complicated. And there are models that various people have built that deal with some of these things. And some of the models are relatively straightforward, and some of them are a little bit more complicated and hard to comprehend. I think that the 98 model worked because it captured some essential features, and it was limited to two stressors, light and nitrogen. For phosphorus, I think that under nitrogen-limited conditions, you have to account for the accumulation of a storage pool. What I didn't show you, I would have more time permitted, was a model that Eric Boutenhouse and I did, which looked at iron limitation and light limitation. It's actually based very much on the way we approached nitrogen limitation, but we substituted iron for nitrogen. And in that, we had to also allow for a storage pool of iron. And Eric has implemented that in their plankton models to look at the effect of that. He's done that. It's a lot easier if you assume that there's a threshold limitation. There's either or rather than a multiplicative.
Can